Rose. How's everybody doing today?
Good.
Oh no, my microphone isn't working the way I thought it would. Anybody heard from Kelsey?
No, I'll contact her.
Okay. As you guys can see, Dr. Dima and Dr. Reynolds are here. This is part of one's normal process of going through promotion and tenure. You get periodically reviewed. Part of that review is that fellow faculty come and watch you teach. So just ignore them in the background as if they're not here. So no word on Kelsey.
She just texted me and said she's having car problems, and so she's dealing with those right now, and she emailed you.
Oh, okay. I was in class until right before I got on here. So I guess the question now is who wants to finish going through the Buckley and Huey paper since that was Kelsey's job and she's not here.
I think technically it was my job.
Oh, I'm sorry. Oh yeah, it was you, wasn't it? Yeah, sorry, brain fart. Yeah, all right, good. We don't need Kelsey for now. All right, let me get mine up. And where were you at, Elizabeth?
So we just talked about the differences between the cold weather instant, like cold spell incidences, and the days that are falling below that 10%.
Yeah, so you're in
baseline temperatures. Yeah.
You're in figure three, right? Yeah, okay, good.
Yeah.
That's where I thought we were. Okay, cool.
So you can see if you go figure three for 3A, we were looking at how the change in the warm spell incidence had increased. And you can see that because in almost in all latitudes actually, because that line is then above the 0% change. And then there was also an increase in number of days above that 90th percentile in the recent years compared to the baseline years. And again, that line, except for at the 80 latitude, is pretty much above the 0 mark, 0% change. And then you can see that in 3B, there's been kind of less of an increase, but still of an increase in the change in cold spell incidence between baseline and recent years. As you can see that like at pretty much 0 degrees, and then more north, there's been an increase, a small increase in cold spell incidences, but not to the same extent as the warm spell incidences. And then there's been a sharp decrease in the number of cold nights that are falling below that 10th percentile temperatures of the baseline years. So you can see that the number of cold nights has decreased a lot in the tropics equator region.
Yes, that is how I read those graphs also. Yep.
Okay. And then.
So before you leave figure 3, and this is to the whole class, when you think about the number of the number of highs in the top 10 percentile and the number of lows that are no longer in the lowest 10th percentile, these kinds of differences are happening mainly where and in what part of the world? Is it happening in tropical regions? Is it happening in temperate regions, or is it happening everywhere equally?
Sorry, can you repeat that question? Just like the first part of it.
Yeah, so when we look at both the TX90P and the TX10P, the number of exceptionally warm days that occur in the higher or in the upper 10th percentile or higher, or days that occur in the lower 10th percentile or lower, are those changes largely happening across the globe equally, or are they changing more in the tropics and less in temperate regions, or more in the temperate regions and less in the tropics? Is the distribution across the globe equally of those kinds of changes relative to baseline?
So for the TX90P, the warm weather days, you can kind of see that it's more of an increase across all latitudes, although there's seems to be a little bit more in the temperate northern region, whereas in the cold nights, there's a sharp decrease in the tropics region, whereas like in the temperate regions, it's staying around that 0% change.
Okay. And when somebody other than Elizabeth, when you look at what's happening in South America, so in the southern temperate regions, you see that both of these things don't really change that much. What was their rationale for why the southern temperate regions weren't being affected by these temperature changes? And the clue to this is by looking at the map to the left in figure 3. What do you notice about the global distribution of land masses?
I can answer this if you want me to.
I want somebody else.
Okay.
Just because you're on the hot seat doesn't mean that everybody else shouldn't have read. Anybody? All right, Elizabeth, do it.
So it's my understanding that they excluded a lot of data from the southern latitudes because there's just not much landmass there. And
yep. So part of it is that they didn't have 10 cells of their grids, especially as you get down into the extreme areas, but even down to about 40 degrees, you once again see that the change isn't really very, very high, especially for TX90 and TX10 or TN10. And so what was their rationale for those areas where they did include data, why you're not seeing much of an effect? What happens to South America as you get further and further south? Relative to its shape and the oceans around it? And now Elizabeth's not even talking to me. What's the southern part of South America surrounded by? Rose, talk to me.
Paul, I'm going to be completely honest.
Yes.
And I didn't want to tell you this way, but my dad died yesterday, and I probably shouldn't even be on this call.
Okay, that's fine. If you're not up for this, that's fine. Elizabeth Quinn.
Can you ask your first question that you asked again?
So when you look at the figure 3 all the way to the right hand side, you see that as soon as you get out of the tropics in the southern hemisphere, if you go down on that figure, the changes from baseline in the number of extremely warm days and the number of cold nights hasn't really changed very much. And there's a good reason for it, and they point this out in the paper as to why those southern temperate regions are not behaving the way the northern temperate regions are. And I was curious as to whether you guys grabbed onto what that reasoning is. And it has to do with the shape of the continents. It has to do with two things. One thing Elizabeth mentioned, which is that there's just not as much data down there in the southern hemisphere. And the other reason is related to something that we've talked about back when we talked about biogeography, but it's also important when we think about why the climate of islands are the way they are, but it also affects continental areas as well. And it has to do with the what's happening to the shape of South America as you go further and further south in South America. Does that ring any bells?
Is it because it's coming to one point, like it's forming like a tip?
Yes, it is forming a tip, and it's a tip surrounded by what?
Ocean.
Oceans. What do you know about environments that are surrounded by oceans? Just as an aside, I used to work in Puerto Rico, and when I would come back from Puerto Rico in the winter, people here at Jewel would be like, "Oh, it must have been so warm there," and I'm like, "Yes, it's very nice there in the wintertime." But when I would come back during the summer, people would be like, "Man, it must have been so hot there," because of course it's in the tropics. But because I was on an island, I would come back in the summertime, and I would have left Puerto Rico, and it was like 82 and sunny. And I would get back here and it's 95 and miserable. What happens when you surround a landmass with a large body of water? What happens to fluctuations in temperature because it's surrounded by water?
Aren't the fluctuations a lot smaller down there?
Do you recall why that's the case, Kiara?
Is it because they're surrounded by oceans?
Yeah, and what does that do to the atmosphere?
Doesn't it, I mean, just because there's ocean bringing in like sea currents and stuff, it just doesn't give as much temperature fluctuations.
So the ocean itself provides a buffer. So if you're on an island and you're surrounded by ocean, the air above that ocean is going to be generally more humid than if you're in a continental region. And they talk about the difference between coastal areas and continental areas in the paper as well. Because you have all of that water in the atmosphere, water has a high specific heat, which means that it can absorb a lot of energy without changing temperature very quickly. And so because you're surrounded by water, you have more water in the atmosphere. that water can absorb a lot of energy without changing temperature. And so you tend to find these places without a lot of temperature variation. Well, that works for islands, but it also works for continents and portions of continents that are right next to oceans. And so as you get down to the southern hemisphere, you find that the oceans are next to that continent at a place where it's thin enough, there's not as much of a continental landmass there, that the oceans themselves can buffer those temperatures. And so you're not going to see the same kinds of changes in the southern temperate regions rather that you see in the northern temperate regions. And they also talk about one of the reasons that the northern temperate regions do fluctuate so much is that you have big continents there. If you get far enough into the continent, the effects of those oceans don't really have much of an effect on climate. And so you see higher daytime temperatures, colder nighttime temperatures, because you don't have that buffering of all that humidity in the air. All right, Elizabeth, continue.
So if you move on to figure 4, they're showing the species richness of different taxon. Graph A shows their ant or insect species richness, and then B shows reptiles and amphibians. D is for the birds, and then E is for mammals. And so I thought figure A was the easiest to understand and read, just by the way they depicted the critical thermal limits versus the lethal thermal limits. And they also had like the strongest slopes of the lines for this one. So you can see what they have on the overlaid on all of them is the average temperature and then the mean annual maximum and minimum temperatures, which is the Tx sub x and Tn sub n. And so we kind of talked about this in terms of like optimal thermal physiological performance. So you can see there's a lot more variation at the minimum temperatures for these different ant species. Whereas at the higher temperatures, there's pretty much a very strict limit at that kind of 40 to 50 degrees Celsius at the right side of the graph. And so what I took from figure 4A was that the ants kind of have the widest, broadest temperature ranges, and compared to the other taxon that they examined.
And of course, who has the narrowest?
This is kind of confusing for me. It seems like the amphibians and reptiles, but then
well, the amphibians and reptiles look to me they're still pretty broad across that range once again, but look at the birds and mammals.
It'd be the birds and the mammals and especially the mammals, I guess, especially in that like 20 to 20 degrees latitude range, where they prefer like being warmer than the average temperature overall, generally speaking. But then they also have a much broader latitudinal range than the amphibians and reptiles into that northern latitudinal region.
Yeah, so this is for anybody in the class. Why are the birds and mammals behaving differently in terms of the way these graphs are drawn in terms of what the thermal environments are like where they're at versus what their thermal physiology is like compared to the amphibians and the insects? What's the fundamental difference between those two groups? birds and mammals versus amphibians, reptiles, and insects.
So the birds and mammals should be endothermic, and that should be like affecting like how much energy they need as well as when they're active. So I think they mentioned that the birds and mammals are way more likely to be nocturnal in that tropics region as a way of avoiding daytime heat.
Yep.
Yeah.
Yeah, so they're creating metabolic heat production. So they're going to be maintaining their body temperatures at generally higher temperatures than amphibians and reptiles and insects in particular will be. In addition to that, they also have narrower operating temperatures and preferred temperatures than these organisms that are ectothermic, because they simply have more control over their metabolic heat production, and they don't generally allow their body temperatures to fluctuate a lot. And so, like Elizabeth said, they're going to be moving to nocturnal activity periods in the parts of the world where it's just generally hotter. And this also allows them to occur at higher latitudes than amphibians and reptiles can occur at. So that's the reason that those figures look kind of the way they do. They've basically graphed the ectotherms on one line and the endotherms on another line in that figure, and they did that probably for a reason. All right, take us home. What's kind of the big picture point of all of this?
So, big picture, they were attempting to look at, like overall, they were attempting to look at how biodiversity might change due to climate change, using those two different versions of climate change. And then they predicted that species in the equator would be hit the hardest because there's more species present in the equator. And then there's also a narrower range of temperatures in the equator. So they're going to be less physiologically capable of adapting to those changes. And so in their conclusions, basically they were saying that it was kind of, I guess I can give a short talk about figure 5, but they basically said that they're within or relative and absolute metrics didn't necessarily predict very well the changes in temperature in the recent years. And so that kind of affects their previous graphs, those cold spell, warm spell, and then the absolute temperature changes. And so they kind of basically said that they needed to include more information about, or more climate data, whether that be like vegetation cover, as like we know that affects how organisms interact in their environment and how they can cool themselves. Because they basically said the difficulty in creating these kinds of latitudinal gradient maps was that the scope of how they're examining changes in the climate versus changes in local habitats, local ecosystems is different. And so if that makes sense.
Yeah, I was hoping that you were going to comment on scale. So they're doing this at the level of 1/2 degree latitude and 1/2 degree longitude. That's a huge spatial scale because they're trying to gather these sort of global temperature trends. But even though that's happening at that really large scale, we've talked about back in the chapter that we talked about their temperature relations. You know, these are animals that are moving across really small local scales. You can move out of the shade into the sunlight and raise your body temperature 5 to 10 degrees centigrade. And then when you get too hot, move back into the shade or crawl under a rock and things like that. So they talk a lot in the conclusions and the discussion. They list kind of a bunch of caveats, don't they, as to why their data are maybe not quite as useful or quite as predictive as they could be. And a big chunk of it is this whole issue of scale. They're gathering data at these really broad geographic scales, and organisms themselves are interacting at scales of a few meters in some cases. And so it's difficult then to predict when taking this large scale data and trying to extrapolate that down to this really narrow scale data. But the general conclusion is I think the one that you hit on, which is that even though we think of the tropics as warmer, they're not necessarily warmer as much as they are warmer during some times of the year, but what they really are is they're more consistent and more constant over time, such that as fluctuations increase over time with climate change, those sections of the world are probably going to be more prone to species having problems, running into problems, going extinct and things like this, because they haven't evolved in a fluctuating environment. And so they're going to be more prone to the effects of climate change because it is changing, but the nature of the change is that it's leading to more of these extreme temperature events. And they're just not physiologically equipped to deal with that level of fluctuation. Temperate regions fluctuate a lot because of all the reasons that we kind of talked about earlier today and then last time. As a result, those organisms are well equipped, both behaviorally and physiologically, to deal with those large fluctuations. And so they're predicting that the tropics are where we need to be most concerned, not just because of the way the thermal physiology of these organisms is calibrated to that variation, but also that's where a lot of the species diversity of these groups are. And so that then just has compounding implications for biodiversity conservation. Questions about this paper? As always, is it clearer now than it was when you first read it? Okay, well, that's good. That's to be expected. All right, any questions at all about the paper? All right, then. We'll file that away. The next phase of what we're doing in class is starting to get into the reproduction of amphibians and reptiles. You'll notice in the textbook that they cover reproduction in amphibians in a chapter separate from reproduction in reptiles. And that's because amphibians and reptiles are really fundamentally different in terms of this aspect of their biology. So, can you guys see my screen? Okay, good. All right, so I asked you guys to think about this chapter in terms of constraints and how organisms, how the biology of amphibians place certain constraints on their potential reproductive biology. So I'm going to give you just a little brief introduction here, and then we'll start talking about what those constraints are and what that might mean for amphibians as they go about the process of attempting to reproduce. So, there's a bunch of information in this chapter. So there are both external and internal factors that govern reproductive activity in amphibians, so you should be familiar with those. We'll talk in particular at the beginning of class today about the internal factors, but those internal factors are obviously prompted by external stimuli like temperature, day length, rainfall, stuff like that. There are lots of different modes of fertilization, and these things are taxonomically related to a certain degree. So you will find in your book where they talk about internal fertilization, and they're mainly talking about salamanders and caecilians. And when they talk about external fertilization, they're mainly talking about frogs and toads. There's this cool thing called hybridogenesis and kleptogenesis that occur in some salamander groups within the United States. that are interesting from an evolutionary standpoint, but are also just interesting because you can go over to Northern Illinois and collect some of these individuals. And it's interesting to know what's going on with that. And so we'll take a brief diversion into hybrid organisms and organisms that basically steal other species genomes during reproduction, and then they reject them after they have stolen them. It's an interesting genetic system in some of the Great Lakes area salamanders. So a big chunk of the chapter is talking about reproductive modes. And when they talk about reproductive modes, they're talking about whether or not organisms are oviparous or viviparous, whether they lay their eggs in water or whether they lay their eggs on land and things like this. That leads to a discussion of direct development and viviparity. We've already talked at some length about direct development and viviparity, so we're probably not going to spend a ton of time on that. But there are environmental conditions under which you might expect direct development and viviparity to evolve based on kind of the constraints that amphibians face. And then towards the end, we'll talk about costs and benefits of parental care and costs and benefits of reproduction in general. And you should be able to explain how metamorphosis is controlled. So in those organisms that have larval stages, which is what we have with amphibians, what is the process by which you transform from the larval form into a small version of the adult form? And that's controlled by a complicated group of hormones that have actually been very difficult for biologists to kind of tease apart. Dr. Reynolds, one of Dr. Reynolds' professors, did this, tried to crack this nut while he was a doctoral student, and really ran into a lot of problems with trying to figure out what's going on inside of an amphibian as it goes from a free-living aquatic larva to a free-living terrestrial adult. It's a very, very tricky thing to do. And then the last thing that your book talks about is paedomorphosis, these species that retain their larval characteristics as they achieve reproductive adulthood. So they're reproductively capable organisms that retain a bunch of larval characteristics. We've talked about a bunch of these back when we were talking about amphibian diversity, and we'll go into, once again, what are the selective forces that might have led to the evolution of paedomorphosis. So you should be able, by the end of all of this, you should be able to talk to me fluently about all of these different topics. But what I really want you to know as we go through this is always be thinking about what the environments are that these organisms live in, so that you can relate, okay, why foam nests? If a frog is building a foam nest, what is the gain in exerting more energy and more materials in the terms of things like proteins and whatnot to construct this foam nest, which will then enclose those eggs? Why is it doing that rather than just hopping off to a pond and laying eggs in a pond and leaving them there and then going off about your business, which might be a less costly way of doing reproduction, but there are hazards and things with laying your eggs in a pond. So always be thinking about the environment. So the first thing to think about when we start thinking about reproduction is sex determination. So in amphibians, it's determined chromosomally. And so it's what is known in your book as genotypic sex determination. So you have, just like in humans, we have XX and XY. This is the male in humans, this is the female. This is the heterogametic gametic sex because it has two different chromosomes that make you male in a genotypic sort of sense. In amphibians, both males and females can be heterogametic, but once again, it depends upon the taxonomic group. Female heterogamety is the ancestral condition, so that's generally the rule in amphibians. And so in this case, females are ZW and males are ZZ. And this will become important here in a little while when we talk about things that can change the sex of amphibians. And so, whoa, something happened. Oh, there we go. So the sex of amphibians can be influenced by environmental factors. So we generally think of sex determination being this genotypic sex determination, but there are things that can alter this. And so your book talks about this. Does anybody remember from your book what things they're talking about when they talk about changing sex? And it's generally changing males into females. Towards the very beginning of the chapter.
I'm pretty sure pesticides was like the main one that I know that affects hormone production.
Yes, pesticides are well known for this. So if you recall, this is from back when we talked about reptile temperature-dependent sex determination. And we talked about how gene expression of aromatase and reductase is actually temperature sensitive. And so you begin with testosterone, aromatase converts it into estradiol, which then affects gonad development into a female. And under other conditions of temperature, you have an increase in reductase, which then takes testosterone and transforms it into dihydroxytestosterone, which is the active form of it. Well, this guy named Tyrone Hayes at UC Berkeley has been working with Xenopus levis frogs. They have some introduced populations out in California of this frog. These are frogs that have escaped from the pet trade for the most part. But you'll also recall back when we were talking about the very introduction at the beginning of the semester, we talked about some organisms of amphibians are used as model organisms, and Xenopus levis is one of these. So in these natural populations out in California, some of them occur near golf courses, and these golf courses have used atrazine as a pesticide in the past. And what he noticed back when he was a graduate student actually is that atrazine tends to have a feminizing effect on frogs. So if you have control males, and these are males that are determined by looking at their genotype for their sex chromosomes, control males secrete a certain amount of testosterone, but if you treat these males with atrazine and even as low as 25 parts per billion, they basically lose their ability to produce testosterone. They're essentially no different than control females. In addition to this, they grow ovaries, and he has an entire population in his lab actually that are made up of ZZ males, but he has some of these ZZ males that are control males that grow testes, produce sperm, and cull and do everything that you would expect a male to do. And then he has another set of ZZ males that he has subjected to atrazine, once again at 25 parts per billion. And all of them have ovaries. They do not cull, they do not set up territories. They don't undergo amplexus with the female, and they'll actually produce eggs. And you can mate a ZZ male with a ZZ female, an atrazine influenced genotypic male, and they will produce fertile offspring. And so it's a really kind of cool system to work in, because you can essentially do lots of interesting things with these animals. So he's got a series of papers. So this was published back in 2002. He's got some papers starting around in 2010, where he's actually just working with this single population that is all male, but has control males that exhibit male characteristics, and he has atrazine influenced males that essentially live out their lives as female. And they do have testes, but the ovaries are the larger of the two gonads. And once again, they produce eggs. And they'll copulate with the males and produce viable offspring, which is unusual. But this is all due to environmental pollutants. And what it does is atrazine evidently upregulates the gene that produces aromatase, and that then sends you down the path to becoming female during your development. So what prompts reproduction is generally some sort of environmental stimuli. In most cases with amphibians, it's temperature, day length, and rainfall. All of these things can interact with one another to basically prompt the hypothalamus to produce gonadotropin releasing hormone. Gonadotropin releasing hormone influences the release of luteinizing hormone and follicle stimulating hormone by the pituitary gland. And these two things have different effects in males and females. In females, the luteinizing hormone basically stimulates ovulation, but it also stimulates testosterone production in the cells of Leydig in the testes of males. So that leads to these changes in the gonads, the production of gametes, but then it also leads to the production of other hormones that influence not only secondary sexual characteristics, but also reproductive behaviors. So one of the other things that Hayes has found in his Xenopus is that it not only affects things like the development of the gonads, but it also affects copulatory behavior, competitive behavior when you're competing for territories as a male. And it also leads to changes in the vocal cords. And so the muscles that surround the larynx that are important for generating frog calls in these Xenopus frogs are very different. So he's looked at muscle mass around the larynx and found differences between, for example, control males and atrazine treated males. So it affects lots of aspects of their biology when you come into contact with something like atrazine. He has received a lot of grief from the company that produces atrazine as a result of this. And so there's a whole series of articles that are written about his work that deal with what the company that produces atrazine has tried to do to get him to stop doing research in this area. To no effect. I was just reviewing a proposal from one of his graduate students, and he continues to work in this area. So because temperature and day length and rainfall are important to prompt this cascade of events that brings about reproductive behavior, seasonality is particularly strong in a lot of species, but some species do produce year-round. Eleutherodactylus coqui, which I work with in Puerto Rico, is one of these frog species that tends to reproduce throughout the year. Once again, it lives on an island where rainfall is relatively constant throughout the year and temperatures are relatively constant throughout the year. So we shouldn't at all be surprised if that is the case. But there are a number of species that do not reproduce every year. And your book talked about some of the reasons why some of species tend to not produce every year. They may produce every couple of years, or they may produce at even longer intervals. What are some of the reasons why you might expect some amphibian species to not be as predictable in terms of their reproduction? And this gets to costs of doing reproduction.
Like if the cost of reproduction is really high, if like they're giving birth to like directly developing species like live births, or if they have parental care, if they're like guarding a nest.
Okay, so anytime you're putting a lot of energy into reproduction, and this could be either because you produce a lot of offspring or those offspring are very large, that could be one energetic cost. Another huge energetic cost could be something like plethodonid salamanders that we've talked about before. You lay these eggs on land, they require your protection, so you're spending a significant amount of time in parental care. That time and all of that energy that you spend on parental care may actually prevent you from breeding on a yearly basis. What other things might influence organisms not breeding on a yearly basis? Somebody other than Elizabeth.
Would like climate change have an effect on that?
Okay, explain that for a minute.
Just like the differences from year to year in like higher temperatures or lower temperatures in that specific area.
Okay, can you think of a particular species that we've talked about that may go through long periods of dormancy because of, for example, extreme drought? Some of them occur in Africa and some of them occur here in the United States, out in the western parts of the United States. They have a behavior where they bury themselves down into the soil and they excrete mucus, and that mucus forms a cocoon around them. Do you remember the group?
Basically any of like the spadefoot toads, or like we talked about I think the African bullfrog does it.
Yep. Yeah, both of those species will spend sometimes years without breeding simply because environmental conditions are not suitable. And then when they do get these torrential downpours, they sense those as the water percolates down through the soil. That begins to break down the mucus cocoon that they have formed, and then they'll unbury themselves, go off and breed, and then come back and bury themselves again until it rains again. And in some cases, that's just a series of months, maybe every year. But if you don't get good, for example, out in the western parts of the United States, if you don't get good monsoon rains that come around every year, you may have species skipping a year or two, depending on the environmental conditions. Yeah, so those are the kinds of things that might prompt some of these species to not breed quite as often. So when we think about constraints in amphibian reproduction, when we think about what amphibians are doing in the process of reproduction, we have to always remember that these amphibians lay unshelled eggs. As a result of that, what's going to be the consequence for them in terms of where they can reproduce and when they can reproduce?
There has to be water present.
Yeah, so because you don't have this outer shell protecting your egg, you have to have ways in which you keep those eggs moist long enough for the larvae to develop to the point that they can either hatch out and become free-living larvae in something like a pond or a stream, or they can finish going through direct development and then emerge as little miniature versions of the adult. And so the primary mechanism by which amphibians are constrained is this idea that they're tied to water for their reproduction. And so with that in mind, take a couple of minutes to think about all these different aspects of amphibian reproduction. What's the mode of fertilization? Is it internal or is it external? What's the mode of reproduction? Do you produce eggs or do you give birth to live offspring? The mode of larval development, are you a free-living larval stage or do you go through direct development? In terms of parental care, do you have no parental care? Do you guard your eggs? Do you brood your eggs? Or do you have something like viviparity going on? And then the specifics of egg laying. Do you lay your eggs in water? Do you lay your eggs in foam nests? Do you lay your eggs adhered to leaves and things like this? All of these things can be influenced by the fact that these amphibians are ultimately tied to water for reproduction. But within the diversity of amphibians have found all sorts of ways around being tied to water. So first, look at this list of things and ask yourself, first of all, are these different things independent of one another? If so, if not, tell me why.
So external fertilization shouldn't be, or should be connected to producing eggs.
Okay, so yeah, so obviously if you're viviparous, if you're giving birth to live offspring and you don't have an external egg stage, then obviously all of the viviparous species should have internal fertilization as opposed to external fertilization. What else is not independent? this list.
When I think of like high parental care, you usually think of them guarding their nest or building a nest versus like, I don't really think of live birth and parental care.
Okay, all right. Trying to think the generality of that statement. So when you think about parental care, one of the ways of thinking about parental care would be to think about, you know, sitting on a nest of eggs, something like Ambystoma opacum, the marbled salamander does. They lay their eggs in these depressions in the fall down in southern Missouri and further south. And what they'll do is they'll lay their eggs in these depressions, and the female will sit on top of the eggs and guard the eggs until the spring rains come. So they may lay those eggs in November, and they'll guard those eggs until January or February when we start getting early spring rains in those parts of the world. And at that point, they will then abandon those eggs because those eggs will hatch because they've been developing that entire time that the female has been guarding the eggs. And then they'll leave. So that's one form of parental care. But there's another, so this is the problem with amphibians. You have to think of all the weird things that go on out there. So we have caecilians, for example. And remember that do caecilians have internal or external fertilization? These are those primitive, almost worm-like or snake-like amphibians. I will give you a hint. They have a thing called a phallodeum that you see pictured here. And so these guys have internal fertilization. So when we were talking about caecilians back when we introduced the different amphibian groups, I told you that there are some groups of caecilians where the eggs hatch out inside of the female, and then what do the larvae do as they stay inside of the female?
They basically eat like the lining of the body cavity, or I don't know what it's called, but
they eat the lining of the uterus, yeah. And so in caecilians, they're technically brooding eggs within them, but then the larvae hatch out and they actually feed on the lining of the reproductive tract until they're big enough that they then give live birth to these free living larvae. So that's a case where you have brooding, but you also have one form of parental care, which is basically provisioning these offspring by allowing them to feed off of your reproductive tract. There's another case within caecilians, and I don't know why caecilians do all the weird stuff, but there's another case where the female gives birth to the offspring, and we'll talk about this in more detail when we get to caecilian reproduction, but the female secretes, well, she doesn't secrete, she has skin that becomes thicker, but it becomes thicker because she's developing these lipid glands inside of her skin. And after the larvae are born, she doesn't really guard them as much as she provides her skin as a food source. And so you'll see the larvae feeding off of this the skin that has increased in its thickness because it has all of these lipid glands inside of it, and they're getting energy from eating her lipid-rich skin. Once again, a form of parental care that doesn't really involve guarding eggs as much as provisioning the offspring after they're born. So Yeah, I guess what you said was generally true, except for all of these exceptions, which seems to be kind of the rule. Clearly, direct development. So you can produce free living larvae, but those free living larvae can be produced via oviparity or viviparity. And simply because you are oviparous doesn't mean that you're undergoing external fertilization. Give me an example of some amphibians that produce eggs that undergo internal fertilization, but then they lay their eggs out in water or wherever. So normally we would associate external fertilization with oviparity and internal fertilization with something like viviparity, but we can also associate internal fertilization with oviparity. What's a group that you can think of that does that?
Would it be the tailed frogs?
So tailed frogs are certainly in that category. Who else? That's once again one of the exceptions to the general rule, but I'm thinking of a more general pattern that applies to more species. Kiara, help us out.
Are we talking about just frogs?
No, any amphibians.
Is turtles?
Reptiles.
Oh.
Sicilian salamanders and frogs are the groups that we need to think about. Quinn, any ideas? It's in the next set of slides. Not. Okay, so in the next set of slides, what are these things that we're looking at? We've talked about them. Yeah, these are spermatophores. We've talked about them in the past. You've got this gelatinous platform, and at the top of this gelatinous platform is a big ball of sperm. These are laid down by the males, and the female comes and positions her cloaca over these, plops her cloaca down on top of it, and then basically contracts some muscles that guard the opening of the cloaca, and they'll basically lock the sperm ball off of the top of this thing. And then they store that in a spermatheca. So here is a salamander. This is a figure from Casper's et al. The paper isn't as important as I just pulled the figure from it. But the cloaca is where all of the urogenital substances leave the body of the animal, but she will take the spermatophore up into her cloaca, and then it will store sperm in the spermatheca. And then as the eggs are coming down through the uterus, muscles in the cavities of the spermatheca will basically squirt that sperm over the eggs as the eggs exit. So this is internal fertilization, but this is internal fertilization which involves the laying of eggs. And so they're fertilized right before they're released, rather than fertilized while they're in the female and held for a long period of time. So there are plenty of salamanders that have internal fertilization and produce free living offspring. So internal fertilization is required if you're viviparous, but external fertilization is not required to be oviparous. You can be oviparous being internal or external fertilization. So think of these within the diversity of amphibians that are out there, because there are some combinations that are required, but other combinations that are more flexible as we think our way through these things. So, internal fertilization. Salamanders, most salamanders, but certainly not all of them, undergo internal fertilization. We'll talk about some of the exceptions before it's all said and done. But as I said, the presence of a spermatophore taken into the female, stored How long can females store sperm among the salamanders?
I think in the book it said months.
Yeah, in some cases months. So the plethodonid salamanders in particular, they will breed in the fall but won't lay eggs until the spring. Do you guys recall what the reasoning for that is? Many of these plethodonid salamanders will lay their eggs in streams, in the upper headwaters of streams, but some of them lay their eggs on land, but they all go through courtship and mating on land. And so if they're going through courtship and mating in one environment that is suitable for that, but then they lay their eggs in another environment, they have to store their sperm while they're moving from where they mate to where they ultimately lay their eggs. And in most cases, these plethodonid salamanders, they use pheromones a lot in their courtship behavior, and we'll talk about courtship behavior some chapters down the line. But the pheromone production that they use to communicate during courtship is a set of pheromones that tend to get diluted and washed away by water, and so all of their mating and copulation occurs on land, even though some of them will return to water to lay their eggs. So the spermatheca becomes important for sperm storage in salamanders. Interesting thing about the spermatheca is that females can mate with multiple males. And as it turns out, they can store the sperm from different males in different parts of the spermatheca. And they can actually control which male actually gets to fertilize the eggs. This is a form of sexual selection called cryptic mate choice, where it looks like the female is mating with lots of different males, which means that each of those males should have a shot at fertilizing at least some of those eggs. But because she can control which part of the spermatheca she has stored sperm in, she can actually allow some males to actually do the fertilization of the eggs and exclude other males. And so it results in mating systems where it looks like females are choosing males, even though if you look at copulation success of males, it looks like all the males are doing equally well. And so this is called cryptic mate choice, because it's a type of mate choice that biologists can't see. And we only know this because people who study this have actually genotyped the offspring and found that a female will produce a bunch of offspring that are sired by one or two males, even though she may have mated with 5 males. And they've gone on to find that when they then look at the sperm in different parts of the spermatheca, she's actually choosing, in a sense, which sperm go into which part of the spermatheca, and then she releases certain sperm and doesn't release others. Another cool little aspect of salamander biology. External fertilization in salamanders only occurs in the Cryptobranchidae and the Hyenobiidae. So if you go back and look at the phylogeny of amphibians from back when we talked about amphibian diversity, you will see that these two families are in this basal group of amphibians, the earliest known amphibians to kind of branch off from the rest of the amphibians. All of the rest of these amphibians will do internal fertilization, except maybe the Sirenidae, the sirens, the lesser siren, the ones that have the larval characteristics and the external gills. We don't know enough about them to know what their mode of fertilization is, but all the rest of these guys are all in the internal fertilization. So there is this phylogenetic signal of fertilization mode, at least in salamanders. And we don't really have time to start getting into caecilians and frogs and toads, so we'll just kill this at this point. Questions? All right, I will see the rest of you on Wednesday. Hey, Rose, stick around for a minute. I want to talk to you. And I will see the rest of, not Rose Reynolds, Rose Galletti. And I will see the rest of you on Wednesday. Let me know if you need access. Well, they're gone. Hey, Rose, talk to me. Are you there? Rose? Rose Galletti.
Sorry.
Oh, there you are. Hold
Yeah,
on a minute.
my cat was.
Wait, hold on a minute. I'm trying to do something. Yeah, I'm trying.